Hello, my name is Narelli Narcisco. In this event talk, we'll be analyzing the ethical debate surrounding the use of CRISPR-Cas9, an emerging gene editing technology that has advanced the field immensely in the past decade. This past November, a Chinese scientist, He Jiankui, claimed that he had implanted through in vitro fertilization 2 genetically modified human embryos that were carried to term, giving birth to the first genetically modified human beings, the twin girls Lulu and Anna. The news brought up an outcry both among the scientific community and the public, highlighting the ethical debate that has been going on for years in the genetic editing community. Today, we will analyze the use of CRISPR technology on human germline cells through the 2 of the main ethical frameworks that have shaped Western morals and thought, duty and utilitarian ethics, discussing what are the consequences and arguments that lead to the complex greatness of this field and what is the big picture that we might be missing. Over the past years, the gene editing technology CRISPR-Cas9 has been developed at an extremely high pace. But what is it? The protein and RNA hybrid works as an efficient hunt-and-cut system, where the Cas9 enzyme acts as a pair of scissors to cut the DNA in a precise point. In this point, new genetic information can be inserted, then the DNA repairs itself, either incorporating the new information or turning off the gene if nothing is inserted. The relatively lower cost, simplicity, and potential for this technique to treat a myriad of conditions has propelled the field immensely, to alter the genetic information in cells to become immune to HIV, to re-engineer the pancreatic cells fixing diabetes. It all seems possibly achievable. But if we can help people once they have these conditions, why not just simply remove the genes that cause it before the person is born? It would imply to genetically modify humans, not only in adult cells once they are born, but from the beginning of life in embryos and gamete cells, germline editing, essentially designing babies. Designer babies seem like a topic out of a science fiction movie. What if you could choose how athletic or how smart your kid could be, the color of their eyes or their height? While some may see these enhancements as beneficial, altering human germline implies that the changes are passed down to any future children. So, where does the line between helping people and selfishly deciding on the fate of future generation lies on? This debate and the ethical and social implications has been studied through the main 4 ethical theories that have shaped our society: virtue, right, duty, and utilitarian ethics. In here, we have chosen to focus on the later 2, providing opposing views on germline editing. Duty or Kantian ethics argues that we should act only according to that maxim which we believe should become an universal law, a categorical imperative. Thus, that one should develop their own list of imperatives or duties that they believe society should follow. A straightforward example of a categorical imperative is that of not killing. To illustrate it better, take someone who has the duty to be honest with their significant other. They might say something hurtful because it was their duty. Regarding CRISPR for germline editing, renowned scientists and doctors argued that they have the duty to help people inflicted by genetic diseases. Dr. George Daly from Boston Children's Hospital claims that if he can help his patients via germline genome editing, he has the moral duty to do so. Nancy Wexler, a neuropsychologist from Columbia University who lost family members due to Huntington's disease, defends that if it is in one's capacity to make a difference in the field, they owe it to the public to do so. However, if you look at it from a utilitarian perspective, you would find the opposite. The theory developed by Jeremy Bentham and John Stuart Mills claims that actions are right in the proportion that they have utility, that is, that they benefit or promote happiness to society, and wrong as they tend to produce the opposite of happiness. For instance, analyzing the common ethical scenario where one has to choose to kill 1 or 5 people in the train tracks, from a utilitarian perspective, we should always save the 5 people. On CRISPR, bioethicists, scientists, and the population in general use utilitarianism to argue that if unpredictable negative mutations spread in our society, it can be devastating to future generations. Dr. Eric Lander, a geneticist instrumental to the Human Genome Project, which maps the entire human genetic code, believes we should be more cautious with human germline editing for, from a utilitarian view, the results can impact the entire humankind, as we are essentially artificially evolving ourselves. Now, why are the views so diverging in this field? It is intrinsically related to its societal implications. 4 years ago, during the first International Summit on Human Gene Editing, the scientific community reached a consensus that CRISPR is not well enough understood to be applied to humans yet. This conclusion comes from our lack of understanding about the specificities of genes and their different impacts on the human body, and of the CRISPR technology itself. CRISPR can still lead to off-target editing and mosaicism, which correspond to the presence of random mutations and of cells with incomplete editing, respectively. These can leave the subject susceptible to cancer, other genetic conditions, and annul the effect of the gene alteration altogether. It means that, for instance, in Dr. He's experiment, which aimed to make the twins immune to HIV, Lulu and Anna are endangered of undesired consequences and might not even be immune to HIV, failing its purpose entirely. Moreover, a gene can have multiple functions, even if it's deemed a bad one. For instance, according to Dr. Eric Blender, by removing the HIV-related gene, Dr. He also made the embryos more susceptible to West Nile virus and influenza. All of these present serious risks for the health of newborn twins and to their descendants. Here lies the main implication of germline editing. It will necessarily affect the next generations, meaning that we are starting to control the human evolution, and it is irresponsible to do it without knowing the actual consequences of the technology and the mutation that is being inserted. But while Dr. Hess' case concerns specific instances of health-related modifications, the field of germline editing raises ethical issues concerning all the aforementioned, but also eugenics. Yes, the philosophy of creating a superior human race. This concern disturbed Dr. Verdona, one of the discoverer of CRISPR, so deeply that in her book she described a nightmare where a Hitler wearing a pig mask asked to learn more about her amazing technology. Although unnoticeable by some, that is what designer babies essentially might be to, the creation of a generation that will be deemed superior, smarter, taller, stronger, and prettier, and we are starting to define the standards by which they will be considered superior. It is important to point out that none of these physical or intellectual modifications are possible anytime soon, since CRISPR technology is neither this safe nor this specific yet. Although recently, MIT Technology Review denounced the gene alteration in Dr. Hess' experiment as also related to improved cognitive function. However far away in the future, it is still imperative to consider its possibility. Once the technology is capable of doing so, what will our world look like? Will the social and economic inequality be exacerbated by the existence of engineered and non-engineered humans? Will prejudice shift towards CRISPR and non-CRISPR humans? Or will the technology be available to all humankind? In that case, we may subconsciously select characteristics that will lead us to converge to a homogeneous-looking and genetic population, which might decrease our genetic variability, making humankind more vulnerable to environmental changes that might cause our extinction. The main point tying all these questions is that through germline editing, humans are effectively beginning to control and shape evolution through science. Moral questions arise even faster once we realize that. Do we have the right to play God? Do we have an obligation to future generations to prevent this from happening? What are the ethics of purposely editing and evolving the human species? Do we know enough to do so without leading us to extinction by engineering only characteristics that we deem favorable at this moment in time? Or will the removal of random mutation aspect, key to natural selection, be the doom of humankind? The Second International Summit on Human Genome Editing seems to have eased the fears of the scientific community and the public regarding the designer babies' risks, as they vehemently condemned Dr. He and actively moved towards a rigorous translational pathway between research and clinical trials. According to the statement released by the summit, this pathway must be strict with compelling medical need and an absence of reasonable alternative, adhering to different jurisdictions, policies and standards, and the guidance documents published in the last 3 years, as well as presenting preclinical evidence and accuracy of gene modification. This reaffirmation and consensus that germline research should only be used on health-related treatments, and that would be immoral to determine someone's height or IQ, also seems like an indication that a superior human race is far from reality. Further, they propose the creation of an ongoing international forum to foster broad public dialogue to bring awareness to governments, companies, science societies, and citizens to better understand CRISPR technology. However, it is important to point out that this promise of self-regulation where we are all on the same page has just failed. Dr. Hip broke the international standpoints, in part because they are not really enforceable, and no one had the authority to stop him. Here, it is also important to highlight that there is no significant scientific advance in the birth of Lulu and Anna. According to Kiri Mizunuru, cardiologist and geneticist at the Perelman School of Medicine, there was nothing preventing previous researchers who edited human embryos from doing the same, except their own moral and ethics. Given this precedent, who is to say that in some years there won't be another He who will go rogue and develop a new generation of blue-eyed athletic Einsteins? While this proposition is a bit exaggerated, my argument is that self-regulation can fail under the right incentive, and Dr. He has just proved it. This, in conjunction with evidence that both the Chinese government as well as American scientists might have been aware of his intentions in creating genetically modified humans prompted a hard response that called for a moratorium. In an article published in Nature earlier this month, a new coalition of scientists proposes that every country declares a moratorium, perhaps for 5 years, on scientists trying to create babies whose DNA had been edited. We all believe that we shouldn't be going forward, Eric Lander argues. The scientists stress that they are not advocating for a permanent ban or any restrictions on basic research that could eventually lead to the creation of gene-edited babies in the future. Instead, a global moratorium would allow for the creation of an international framework for how best to proceed responsibly. Even with this firm response, there are those that claim that the uproar over the possibility of designer babies is more out of fear of the unknown than its imminent probability for, according to Penn ethicist Jonathan Moreno, designer babies will never be a reality. In his interview to Penn Today, Moreno argues that there are only a few genetic traits that we can actually manipulate because of epigenetics, the way that the environment enables certain qualities to express themselves. In the BFS seminar on Emerging Technologies in Society last fall, we had Dr. Jennifer Cremings, a specialist on epigenetics, come talk to us. According to her, the epigenetic code consists of heritable changes that do not include alterations to the primary DNA sequence. Dr. Cremings explains that it's the classical argument of nature versus nurture. Thus, our DNA is not our destiny, but it provides us with a range where our characteristics might fall. However, I would argue that even just modifying the range described is already changing human evolution, although not directly leading to a designer baby. Further, as the study of epigenetic advances, we might be able to control the environmental and developmental cues in combination with the DNA sequence and reduce the range to the specific characteristic we desire. Thus, Dr. Moreno's assertion that designer babies will never be a reality is a little too categorical. So, if the possibility of designer babies, eugenics, and the possible impact on the future of humankind as a whole are valid concerns, acknowledged by both the public and the scientific community, why pursue human germline editing? The moral justification does not fall far from Dr. His alleged reasons to performing his trial. Scientists are impelled by compassion to help people. Kant would say that we as a society have the duty to care for each other, but one could also use Kantian ethics to argue that doctors first have the duty to do no harm to people, and that the stakes are too high. A utilitarian can argue that it is too dangerous due to the possible harm for future generations. But one can also use utility to argue that a healthier population will lead to a greater well-being in society. Dr. Doudna, one of the CRISPR discoverers, originally believed CRISPR should not be applied to humans at all. Now, moved by a sense of duty to helping the families, she thinks there is a future for the research, but that it is not time to conduct human germline editing yet. It is a complex and great debate where we must develop our own moral boundaries. So, what can we conclude? Dr. His experiment has made genetically modified humans a reality from which we cannot come back, opening a Pandora of ethical questionings. And while the implications for humankind evolution are vast, as Dr. Doudna says, it is unreasonable to expect scientists not to try to understand how to apply CRISPR to cure genetic diseases which impact and bring suffering to millions of people. The second summit acknowledged this moment in our history, when more and more researchers will try to undertake clinical trials for germline editing, which is why their proposal for a strict pathway and regulation is indispensable for the advance in an ethical and conscious manner. However, I believe that the moratorium coalition touches on a crucial point. This framework must be international and rely on policies. Otherwise, it could lead to uneven policies across nations. The impacts of germline editing in evolution of humans concerns the entire society. So, such decision on its standards should be made in conjunction. In this manner, we would be able to have some control and a conscious agreement on where the future of humanity is headed. Finally, while the use of germline editing in humanity is deeply important for all the reasons discussed in this talk, I would like to close with an interesting thought. In the big picture of the field, germline editing of different species, animals, insects, plants, can have the same effects on their evolution, dictating their future or the absence of it. CRISPR technology is already being applied in the germline editing of species of plants and insects, which are being inserted into our ecosystem, and we don't know the consequences that this might have on the environment on the long term. So, there might be a more pressing ethical dilemma that must be addressed as soon as possible.